To express a gene means to transcribe and translate it, making the protein it codes for. All of your cells have all of your DNA, and therefore all of your genes. But obviously these genes are not always being expressed. The cell that makes up your stomach do not express the gene that codes for eye color, and your eye cells don't express the gene that codes for enzymes in your stomach that break down protein. So how do cells know which proteins to express and when? Keep watching and you'll find out. By the end of this lecture, you should be able to describe the functions of genes that regulate gene expression and compare and contrast gene expression in prokaryotes and eukaryotes. Just to give you a little perspective, let's start with a single cell. If we move into the nucleus of the cell, there we will find all of the DNA, all 6 billion base pairs of DNA that makes you, you. Zooming in on a section of that DNA is called a gene. One gene contains several important regulatory regions, or sections of DNA near the gene that turn it on or turn it off. When you hear about a gene being turned on, that means transcription and translation is taking place to produce the protein the gene codes for. When it's turned off, no transcription or translation is taking place. Special sequences before structural genes, or the genes that actually code for the protein, give instructions about whether the gene should be turned on or off. Genes in prokaryotes, or organisms without a nucleus like bacteria, are regulated differently than eukaryotic organisms, or organisms that do have a nucleus like plants and animals. In a prokaryote, there are 4 basic regions for every gene. The first is the gene itself that actually codes for the protein. This is called the structural gene. The next 2 sequences of DNA act like on and off switches. The on switch is the segment of DNA that attracts RNA polymerase so that transcription can begin. This is called the promoter. The off switch has 2 parts, a protein that acts like a roadblock and the place that this roadblock protein attaches. The repressor is a section of DNA that codes for a protein that acts as the roadblock. And the operator is the section of DNA that the repressor protein will bind to. I know this is very detailed and maybe hard to wrap your head around since it's something you can't see, so let's take a look at an example. Milk sugar eating bacteria, the kind that gets in your milk carton and makes it stinky and gross taking. To break down the sugar lactose, bacteria need several enzymes. Each enzyme is coded for by a separate protein, but they all work together and need to be turned on at the same time, so they are all controlled together. A group of genes that are expressed together are called an operon. Operon are only present in prokaryotes. The group of genes that are needed to break down lactose are called the lac operon. So let's see how this works. When those nasty little bacteria are floating around in the air, they're not eating your milk, so they don't need the enzyme to break down lactose. The cell can detect that lactose is not present, and so it sends a signal that causes the repressor protein to be transcribed and translated. In the picture, that's this little pink circle. The repressor protein will bind to the operator, the segment of DNA marked O. RNA polymerase, to be honest, is a little out of control. It wants to transcribe every little bit of DNA that it can, but it has to attach to the DNA and it has to create mRNA in the 5' to 3' direction. So the repressor protein just needs to position itself, what is called upstream, or before the gene, and blocks RNA polymerase so that it can't pass go, it can't collect $200, and it can't transcribe the gene. Why waste energy on transcription and translation if there's no milk to eat? But let's change the game. Victory, those stinking little bacteria made it into the milk carton. Bacteria heaven, more food than it knows what to do with. Now it needs to start making all those enzymes that code for, it needs to make all of those proteins that code for the enzymes to break down lactose. A molecule of lactose binds to the repressor protein, which makes the repressor protein lose its grip on DNA, clearing the way for RNA polymerase. RNA polymerase will attach itself to the promoter region of the gene and begin its work. The promoter is a segment of DNA that acts like a signal to tell RNA polymerase where to begin. Eukaryotes are different. Because they have more DNA and are more complicated organisms, the control of their genes is more complex. But don't worry, we'll just learn a few general principles. Principle number 1. A strand of mRNA may code for several different proteins. But how does that happen? There's one little step of protein synthesis that we left out until now. In between transcription and translation, mRNA can be edited. Introns are parts of a gene that are parts that the cell doesn't need, and exons are parts that the cell does need to make a particular protein. Editing mRNA is sort of like a cut and paste process. Introns are cut out and exons are pasted together. Depending on which introns are cut out and which exons are pasted together, a piece of transcribed mRNA could code for more than one protein. Principle number 2, eukaryotic genes are not regulated in groups. All of the enzymes that are needed to break down lactose have just one repressor protein and one operator sequence and one promoter sequence. While eukaryotes need many different enzymes to do a common job, these genes are usually not all right next to each other like they are in prokaryotes. There are 3 important sequences that regulate gene expression in eukaryotes: the promoter, the enhancer, and the Tata box. The promoter has the same function in prokaryotes and eukaryotes. It's where RNA polymerase begins its transcription. In eukaryotes, a sequence called the Tata box helps to point out to the RNA polymerase where the promoter is. The enhancer sequence is a place on the DNA where special proteins called transcription factors can bind. A transcription factor is a general term for proteins that help to regulate gene expression. Some transcription factors block RNA polymerase, just like the repressor protein does in prokaryotes. But the enhancer sequence could also bind other transcription factors that uncoil DNA, code for proteins that speed up transcription, or other functions. Different transcription factors could have several different jobs. One of the processes that gene regulation controls is how cells differentiate during development. Every organism that's produced by sexual reproduction begins as a single cell, and as those divide, they become specialized or differentiate into specific types of cells, like nerve cells, stomach cells, and muscle cells. These types of cells have different functions, and so they also have different structures, so they look different. Most animals contain genes called Hox genes that control the process of development. They're considered homeobox genes because one set of genes controls the expression of other genes. It works sort of like a cascade. The first Hox genes to turn on control whether the head region develops or the tail region develops. Then the head region genes might turn on arm genes, and the arm genes might turn on finger genes, and the finger genes might turn on the pinky finger versus the thumb. We share the same Hox genes with many different animals, which is why human embryos start out looking so much like fish, turtles, and salamanders. As our cells differentiate further, Hox genes specific to humans are turned on. Some of the last Hox genes to turn on in humans are the Hox genes that differentiate between our thumb and our pinky. Hox genes that are last in the cascade affect development least because they turn on fewer or no other genes. Because we don't usually see people born with no arms, we can infer that Hox genes needed for development are vital. So now that we're done, make sure that you go back and take the quiz. So let me pull up a preview of the quiz for you. Here we go.